Good morning. My name is Christian Howell. I'm the CEO of Cognito Therapeutics. At Cognito, we have a commitment to make sure that we always keep those that are inflicted by neurodegenerative diseases front and center. So I wanted to start the presentation with the story of Sandra Day O'Connor. As many of you know, Justice O'Connor was the first female justice to join the Supreme Court, to be appointed to the Supreme Court. She shaped law in the country for almost 30 years. Unfortunately, in the last chapter of her life, she took on a challenge that was probably one of the few in her life she couldn't beat, which is Alzheimer's disease. In 2007, Justice O'Connor stepped down from the Supreme Court to take care of her husband, John. John had been diagnosed with Alzheimer's disease several years before. When Justice O'Connor stepped down, John didn't know who Sandra was. He had been moved into a memory care clinic. He had fallen in love with someone there, and Sandra knew that this was a part of the disease, that she knew she was going to lose the loved one that she had, but she was not, she still wanted to be there to support him. Then in the cruelest of twists, in 2018, Justice O'Connor was diagnosed with Alzheimer's disease. She died in 2023 from Alzheimer's disease. She died probably not remembering John, not remembering her family, not remembering the impact she had on the United States. And sadly, that story is not unique. So there are 7 million people in the United States that are suffering from Alzheimer's disease. They have lost memory, they have lost function, their loved ones have watched them as they've gone through this, and they've been helpless in accessing any therapy that could help modify the disease. And so at Cognito, we asked, what if that didn't have to be true? What if we could provide a non-invasive, disease-modifying therapy that patients could use in their home that would be effective at not just slowing, but maybe stalling the progression of that disease and could allow them to stay with their families, stay with their loved ones? So let's talk a little bit about how we got there. So we get to stand on the shoulders of giants at Cognito. There has been 30 years of research done by our founders at MIT and Harvard and Stanford that have really answered 5 critical questions, which is 1, is an active brain a healthy brain? And what they have proven to us is yes, that neurons that fire together wire together, and it increases neuroplasticity the more active we are. And we know this. This is conventional wisdom. We tell our parents all the time, stay active. The next question they asked was, what happens biologically when we're active? And the answer became very clear through studies that 4 key things happen. The first is when we are most active, and we consider that to be at the 40 hertz level, that we see synaptic genes and proteins, myelin genes and proteins expressed. We see the lymphatic system, which is the filtration system of the brain, keyed. And then we see a peptide called the vasoactive intestinal peptide expressed, which helps with vascularity in the brain. So then the question was asked, okay, what happens in a neurodegenerative brain? Are they able to reach that level of activity? And the answer was no, that when you have neurodegenerative disease, be it Alzheimer's or MS or Parkinson's, that you have neuronal breakdown, you don't have the ability to drive that level of neuroplasticity, and therefore you can't drive that biologic expression. You lose white matter for gray matter, and you see the death of synapses, so you lose cognition and function. Then there was this really interesting question, which is, could we use an external stimulant to drive 40 hertz? And this was pioneering work by Carl Dieserhoff and Ed Boyden at Stanford and then MIT that's used optogenetics to pinpoint specific genes and proteins to drive these oscillations back. And then the last question, which was really the most pioneering and led to the formation of Cognito, was, could you use sound and light to drive this level of gamma response? And the answer was yes, that by using the sensory on-ramps of the brain, that you could get directly to the auditory and the motor cortex, you could get to the corpus callosum, you could get deep inside the brain, and you could very safely and in a non-invasive way drive these oscillations and then drive this biologic expression. That then drove the formation of Cognito, and Cognito has continued to explore that science in partnership with MIT and Stanford and Harvard, and we have now moved into human studies. What you didn't hear me say in that was amyloid or tau or any of the things that we are currently aligning to Alzheimer's disease. What we're talking about is brain health. We're talking about driving neuronal plasticity, driving myelin, driving the lymphatic system. And so we believe that this science and this technology is not only impactful to Alzheimer's patients, but to an array of other neurodegenerative conditions, to mood conditions, to traumatic brain and concussion, and other areas where we think strong brain health is important. Let's talk a little bit about the mechanism of action. It's going to feel to you like we're a little bit of a tweener, right? Are we a medical device? Are we a pharmaceutical? Are we a therapeutic? We're kind of all of those. To give you a sense of the device, you saw a picture earlier. We use audiovisual stimulation, accessing the optic and auditory nerve. We drive 40 hertz gamma stimulation in the brain. Patients sit with it for an hour a day, every day, and they can do it in the safety of their home. I'll go through the clinical outcomes here in a second, but we hear from patients post our surveys in our UX research that it's meditative, that it's safe, that it's comfortable, that it's easy to integrate into their daily lives, which is unbelievably important for patients with Alzheimer's disease. From a mechanism of action perspective, it's really interesting. We actually have these three very clear biomarkers and then a measurement of a clinical marker. So first, every patient that comes in, we put on an EEG, we put that patient on the stimulation, we turn it on, and we confirm that we see a gamma response. So day 0, we know the patients are seeing a gamma reaction. At 60 days, in our phase 2, we looked at spinal fluid, and we saw this massive expression of synaptic and myelin proteins. So biomarker 2, that we knew that evocation and stimulation was now driving biology and proteins. At 3 months, we looked at MRI, and we saw not just a preservation of myelin, but in some patients, a regeneration of myelin. And then at 6 months, we did the clinical tests. We used the mini mental state exam, and we used the ADCS-ADL, the activities of daily living, and we saw effects that have not been seen in any space before and beyond anything we've seen in biochemistry, and I'll share those with you now. We have been robust in our clinical research. We are currently in our 5th human study. So you see here that we started with Flickr, which was very much a proof of concept. We then did a feasibility in Overture, which was a 6-month randomized, sham-controlled study on 74 patients. We did an open label extension on that of 12 months. That extension showed durability of treatment. It also showed that we were able to arrest the decline in the sham group. We are currently 610 patients enrolled in a 670 patient, 12-month sham-controlled randomized phase 3. It is the largest study ever done in the neurotech space. And so we are incredibly excited. We expect, I'll go through the timeline here in a second, but we're incredibly excited about the size of and the evidence that we'll have moving forward. What did we see in Overture? We saw a 76% preservation in cognition, a 77% preservation in function, and a 69% preservation in brain volume measured through MRI. Obviously, the thing we hear more often than any is it almost seems too good to be true. And I would understand that if we didn't have the 30 years of research behind us and as much robust clinical evidence as we do. These therapies, and I know we also are only as breakthrough or can make as big a breakthrough as if they can get to the patients. And that is not something to take lightly in a medical device, and this is where we now cross over from a therapeutic to a device. But we believe we're in an unbelievably advantaged position. 1, we are breakthrough, de novo through the FDA. 2, we have our letter of intent in on TSET. 3, we have data on over 700 patients in a Medicare population, so we've begun parallel review with CMS. We are incredibly well received by the payers. We are about a quarter of the total treatment costs for the monoclonal antibodies. We'll have about 6 million patients under our label. We can be prescribed by neurologists, primary care physicians, geriatricians, neuropsychiatrists. So the ability for us to gain access to all of those patients that are in desperate need of this therapy is very clear. In closing, I just want to give everyone a sense of the timeline. We will be last patient enrolled here in the next 60 days. We will see last patient out in May of 26. We will release our top line data in July of 26. We'll submit a regulatory approval at the end. And then we're hoping, fingers crossed, for a regulatory approval at the beginning of 27. And then we really do believe it's an opportunity for us to make a massive impact in Alzheimer's disease. So I appreciate everyone's time and attention. Thank you very much.